So here's something worth thinking about as you settle in tonight. Right now, as you lie here letting your eyes grow heavy and your thoughts begin to soften and drift, your body is preparing to do something that looks, from the outside, like almost nothing. You'll go still, your eyes will close, your breathing will slow, and to anyone watching, it will seem like you've simply stopped, like you've pressed pause on the business of being alive. But you haven't, not even close. What's actually about to happen, what happens every single night, in every human being, all across the world, in every culture that has ever existed, is one of the most elaborate and purposeful biological programs in nature. While you lose consciousness, your brain will cycle through at least 4 or 5 distinct states, each with its own electrical signature, its own chemistry, its own specific jobs to do. Your growth hormone will surge to its daily peak. Your immune system will run a kind of overnight maintenance program. Your brain cells will physically shrink, and the fluid surrounding them will flood in and wash out the metabolic waste that accumulated while you were thinking. Your heart rate will slow, your core body temperature will drop by almost a degree. And somewhere in the deep architectural stages of your sleep, the events of your day, the faces you saw, the things you learned, the conversations you had, will be quietly processed, filed, and consolidated into the long-term structure of who you are. And you will feel none of this happening. You will be, for all practical purposes, gone. And when you come back, in the morning, none of it will have left any trace in your awareness, except that you'll feel, if you've slept well, restored, repaired, like yourself again. That restoration is the point. Sleep is not the absence of waking. It is a different mode of operation, active, organized, essential. And for most of human history, nobody knew this. People thought sleep was basically what it looked like from the outside, a pause, a rest, an interruption of the interesting business of being alive. It took until the middle of the 20th century, and a graduate student working the night shift in a sleep laboratory in Chicago, watching a sleeping child's eyes, for science to discover that sleep was something else entirely. That's what we're going to talk about tonight. Everything your body does while you sleep. All of it. The electrical waves your brain generates and what they're for. The hormones that pulse through you in the dark. The extraordinary process by which your brain cleans itself every night. The paralysis that keeps you from acting out your dreams. The hallucinations at the edge of sleep. The strange things that happen to your temperature, your heart, your breathing, your immune system, your memories. We're going to take it slow. Start at the very beginning. Walk through the night together. No rush. If you drift off somewhere along the way, and you probably will, that's not just fine. That's the point. You'll be demonstrating, in real time, exactly what we're talking about. You don't need to be a neuroscientist to follow this. You really don't. Because sleep at its heart is about something you already understand. The need to reset. The need to process. The need to repair. The need, after everything the day has asked of you, to be rebuilt. What we're going to do is just look, slowly and carefully, at how that rebuilding actually happens. At the machinery beneath the unconsciousness. At what is quietly, competently underway in you right now. Let's start where the science starts, which is not with sleep itself, but with a confession of how thoroughly it was misunderstood for almost all of recorded history. Aristotle, in the 4th century BCE, wrote a short treatise on sleep. It is, in most respects, completely wrong, but it is wrong in a way that reveals how intuitive and persistent the wrong idea was. Aristotle thought sleep was caused by vapors. Specifically, he thought that digestion produced warm, moist vapors that rose from the stomach to the brain, and that these vapors, accumulating in the head, caused a kind of heaviness, and torpor that we experience as the onset of sleep. As the vapors cooled and sank back down during the night, the brain gradually cleared, and you woke up. Sleep, in this view, was a kind of metabolic hangover from digestion, a passive consequence of eating, something that happened to you, not something your body was actively doing. This view, or variations of it, dominated thinking about sleep for roughly 2,000 years. The details changed, the vocabulary changed, but the underlying assumption was constant. Sleep is what happens when the brain's activity stops, or slows, or is suppressed. It is the absence of wakefulness, the void between days. If you think of wakefulness as a candle flame, then sleep, in the Aristotelian tradition, is what you get when you blow out the candle. Nothing. Dark. Quiet. A pause until the flame is relit. The assumption was not unreasonable given what was observable. A sleeping person doesn't respond to stimulation the way a waking person does. Their movements cease. Their expression goes slack. They are, to all appearances, temporarily absent from the world. If you had no way to look inside the sleeping brain, no way to measure its electrical activity, no way to watch its chemistry change, no way to observe what was happening at the level of cells and circuits, the sleep as absence model was the only one available. It fit the evidence. It just happened to be completely wrong. The first crack appeared in 1924 in a psychiatric hospital in Jena, Germany, in the laboratory of a physician named Hans Berger. Berger was obsessed with a question that most of his colleagues thought was either unanswerable or not worth asking. Could the electrical activity of the human brain be measured from the outside, through the skull? He had been convinced for years that the brain produced electrical signals, this was known, and that those signals might carry information about mental states. He built his own instruments, refined his own techniques, and in 1924 he achieved what he had been working toward for nearly 2 decades, a recording of the electrical activity of a human brain, traced as wavy lines on paper. He called the recording an electroencephalogram, an EEG. Berger was not immediately believed. His results were greeted with considerable skepticism, and he spent years defending his findings against colleagues who thought the electrical signals he was recording were artifacts, noise from muscle movements, from the electrodes themselves, from vibrations in the room. It wasn't until 1934, when the British physiologists Edgar Adrian and Brian Matthews independently replicated his results, that the scientific community accepted that what Berger had recorded was real, and then everything changed. Because what Berger found, even in his early recordings, was that the sleeping brain was not electrically quiet. It was not a candle flame blown out. It was producing electrical oscillations, organized, rhythmic patterns of voltage that were different from the patterns of waking, but not quieter. Not less, different. The brain during sleep was doing something organized enough to produce characteristic wave patterns, something that changed as sleep progressed, something that was not, in any meaningful sense, the absence of activity. Berger died in 1941 by suicide in the middle of World War II, having been forced into early retirement by the Nazi regime that had taken over Germany. He did not live to see the full implications of what he had discovered, but the tool he built, the EEG, became the instrument that would, over the following decades, reveal the true nature of sleep. Layer by layer, discovery by discovery, the sleep laboratory would show that the brain at night is not a darkened room, but a theater with a changing program, different acts, different scenes, different players, cycling through the night in a pattern as organized and as necessary as anything the brain does while awake. The next great discovery came in 1953. in a sleep laboratory at the University of Chicago, and it came about because of a graduate student and a child. The graduate student's name was Eugene Aserinsky. He was 30 years old, a father, running out of money, and working in the laboratory of a physiologist named Nathaniel Kleitman, who had been studying sleep for decades. Kleitman had an old, cranky piece of equipment, a polygraph machine capable of recording multiple physiological signals simultaneously, and he had been using it to study the eye movements of sleeping subjects, because he had a theory that slow rolling eye movements at the onset of sleep might be related to the depth of sleep. Aserinsky was assigned to collect the data. It was painstaking work, attaching electrodes to subjects, running the machine through the night, interpreting the rolling spools of paper that came out. And one night, watching a recording, he noticed something odd. His subject, who may have been his own young son, Armand, though the exact details are disputed, showed a burst of rapid eye movements during what should have been the quietest phase of sleep. Fast, jerky eye movements, back and forth, precisely the kind of eye movements you make when you're awake and scanning the world. But the subject was clearly asleep, and the brain recording showed a pattern that, paradoxically, looked almost like waking. Aserinsky reported what he'd found to Kleitman. Kleitman's initial reaction was skepticism. He suspected equipment malfunction. They checked the equipment. It wasn't a malfunction. They ran more subjects. The same thing kept appearing, periods of rapid eye movement with brain activity that looked like waking during sleep. These periods were not random. They occurred at roughly 90-minute intervals through the night, each one lasting progressively longer as the night went on. And when Aserinsky and Kleitman woke subjects during these periods, the subjects reported vivid, detailed, narrative dreams almost every time. When they woke subjects outside these periods, the reports were sparse, fragmentary, or absent altogether. They had discovered what we now call REM sleep, rapid eye movement sleep, and they had discovered, in the same stroke, that the night is not uniform, that sleep has architecture, that it cycles, that it contains within it a state so electrically active, so physiologically peculiar, so associated with the most vivid experiences of the sleeping mind, that it represents something genuinely new, something nobody had known was there. Aserinsky went on to have a complicated relationship with his most famous discovery. He published relatively little after the 1953 paper, moved away from sleep research, and in later life expressed some ambivalence about the work that had made him historically significant. He died in 1998 in a car accident at the age of 77. Kleitman, his mentor, lived until 1999 and died at 104 years old, the oldest person ever to have been a Fellow of the American Physiological Society. He had spent his entire career thinking about sleep and had seen it transform, within his own lifetime, from a subject of occasional physiological curiosity into one of the most active and consequential fields in neuroscience. Nathaniel Kleitman had been interested in sleep since the 1920s. He had conducted early sleep deprivation experiments on himself, spending periods of up to 180 hours without sleep in the name of under 50s. There is another figure in the early history of sleep science who deserves mention, William Dement. Dement was a medical student at the University of Chicago in the early 1950s, who joined Kleitman's laboratory and became fascinated by the new phenomenon of REM sleep. He spent years mapping the architecture of sleep in detail, quantifying the cycle lengths, the proportions of each stage, the changes across the night, and then spent decades building the field of clinical sleep medicine, essentially founding the concept of sleep disorders as a medical specialty. He established one of the first sleep disorders clinics at Stanford in the 1970s and spent the rest of his long career. He died in 2020 at age 91, trying to convince the medical establishment, the public, and anyone who would listen that sleep deprivation was a serious public health crisis. He coined the term sleep debt to describe the accumulated deficit from chronic insufficient sleep, and he argued persistently that most Americans were living in a state of chronic partial sleep deprivation without being aware of it. He was right. The gap between how much sleep most adults need, roughly 7 to 9 hours, and how much they actually get, has been widening for decades. The average American in 1942 slept about 7.9 hours per night. By 2013, that had dropped to about 6.8 hours. Over a lifetime, the difference is enormous, roughly 365 hours, more than 15 full days, of lost sleep per year, compounding its effects across every biological system we've been talking about. Standing what sleep deprivation did to the body and mind. He had spent 2 weeks underground in Mammoth Cave in Kentucky, living in artificial light, trying to determine whether human beings had an intrinsic 24-hour rhythm, or whether our cycles were driven entirely by external light cues. He and his assistant, Bruce Richardson, found that both men maintained roughly 24-hour rhythms underground, though Richardson's rhythm drifted somewhat, evidence that humans have an intrinsic biological clock that runs on approximately, but not exactly, 24 hours, and that needs daily light to stay synchronized. This was one of the earliest empirical demonstrations of the human circadian rhythm, though the term circadian, from the Latin circa dies, approximately a day, had not yet been coined. The paper Aserinsky and Kleitman, published in 1953 in the journal Science, is one of the most important papers in the history of neuroscience. It is short, just 2 pages, and written in the measured language of scientific reporting, but what it describes is a revolution. The night was not empty. The night was not a pause. The night was a theater, and REM sleep was its most dramatic act. So let's talk about what that theater actually looks like, because the architecture of a night's sleep, the sequence of stages, their timing, their proportions, what each one does, is the foundation for understanding everything else that happens while you're unconscious. If you were to monitor your brain with an EEG tonight and record every minute of your sleep, and then plot the results on a graph called a hypnogram, a visual representation of sleep stages over time, what you'd see would look like this. You'd begin in wakefulness. Then, as you drifted off, the trace would descend, first into stage 1 sleep, then stage 2, then the deepest stage, slow wave sleep. It would linger there for a while. Then it would rise back up, and instead of returning to wakefulness, it would enter a branch, REM sleep, a flat line at a level different from the deep stage, with a characteristic signature, and then the cycle would begin again, down into deep sleep, back up, another REM period, and again, 4 or 5 times through the night, each cycle roughly 90 minutes, each one slightly different from the last. The first cycles of the night contain more slow wave sleep, the deepest, most restorative stage. The last cycles before you wake contain more REM, longer, more vivid REM periods. If you're cut short in the morning, you lose REM. If you're cut short at the beginning, you lose slow wave sleep. They serve different purposes, and losing either one has different consequences, which is why the quality and duration of sleep matters, in ways that are specific to which stages you're shortchanging. Stage 1 is the entrance, the threshold, the place where wakefulness ends and sleep begins, though the boundary is more gradient than wall. Your eyes, if someone could watch them under closed lids, begin to make slow, rolling movements, not the rapid dart of REM, but a slow, lazy drift. Your muscles begin to lose tone. Your thoughts, if you could observe them from outside, become fragmented and hypnagogic, disconnected images and half-formed sentences that feel coherent in the moment and nonsensical when you try to remember them afterward. Your brain is generating theta waves, slower than the beta waves of alert wakefulness, faster than what's coming next. Stage 1 typically lasts just a few minutes. It is a corridor, not a room. Stage 2 is where you spend most of your night. Roughly half of total sleep time in a healthy adult is Stage 2. The EEG trace in Stage 2 shows two distinctive features. The first is sleep spindles, brief bursts of electrical activity, 11 to 16 hertz, lasting about a second, that look like little spindles on the paper trace. They occur about every 20 to 30 seconds and are produced by an interaction between the thalamus, the brain's relay station, and the cortex. The second is K-complexes, large, slow waves, like a sudden sharp dip, and rise in the otherwise rolling trace. Both features appear to be related to the suppression of arousal. The brain, in Stage 2, is actively managing its own level of consciousness, damping down responses to external stimuli that might otherwise wake you. A noise in the house, a car outside, a shift in temperature, Stage 2 sleep spindles seem to protect you from waking up to these things. The brain is choosing to stay asleep. It is a choosing, not a falling. Stage 3, which used to be divided into stages 3 and 4, but is now classified as a single stage, is slow wave sleep, or deep sleep, or delta sleep, named for the slow, large amplitude delta waves that dominate the EEG trace. Delta waves are less than 4 hertz, fewer than 4 cycles per second, and they are the slowest, highest amplitude waves the sleeping brain produces. The brain in slow-wave sleep is oscillating in a stately, synchronized way, like a vast chorus all breathing together. Individual neurons are cycling between periods of firing and periods of near-complete silence, over and over, in a rhythm that propagates across the cortex in coordinated waves. It is, electrically, the most organized thing your brain ever does, and it happens while you are the least aware. Slow-wave sleep is when the body does its most intense physical maintenance. This is also when the kidney, as we discussed, is running in its most conservation-oriented mode, holding on to water and concentrating waste. It is when the liver's metabolic rate is lowest. It is when the gut motility is reduced and digestive processes enter their quietest phase. The entire metabolic rate of the body is at its nadir during slow-wave sleep, with one major exception: the immune system, which is actively upregulated, and the cellular repair machinery, which is running at full capacity. It is as if the body, in deep sleep, has redirected the energy that normally runs perception and cognition into the maintenance and repair of its physical infrastructure. This is when growth hormone is released. Roughly 80% of the daily growth hormone secretion happens in the first slow-wave sleep episode of the night. Growth hormone is often thought of as the hormone that makes children grow, which is true, but its role in adults is equally important. It stimulates protein synthesis, drives tissue repair, mobilizes fat for energy, maintains muscle mass, supports immune function. Every night, during slow-wave sleep, your pituitary gland releases a large pulse of growth hormone into your bloodstream. Your cells respond. Damage from the day is repaired. Worn proteins are replaced. The body rebuilds itself, quietly, in the dark, while the EEG traces its slow, deep oscillations. And this is also when the brain does something that was discovered only in 2013, and that may be one of the most significant findings in neuroscience of the past decade. Let's take a moment with it, because it changes how you think about what sleep is for. The brain, while it is awake and working, produces waste products. This is not metaphorical. It is literal chemistry. Neurons firing, neurons communicating, neurons doing the work of thought and perception and memory. All of this produces metabolic byproducts, just as any active tissue does. And some of those byproducts are proteins, amyloid beta, tau protein. In healthy brains, these proteins are cleared away efficiently. In brains where the clearance system fails, as it fails in Alzheimer's disease, these proteins accumulate into plaques and tangles that disrupt neural function. The accumulation of amyloid beta and tau is the defining pathological feature of Alzheimer's. They are, essentially, the brain's garbage. And in Alzheimer's, the garbage doesn't get collected. For years, researchers knew that the brain produced these proteins and knew that they needed to be cleared, but they didn't understand how. The brain has no lymphatic system, the network of vessels that clears waste from other tissues throughout the body, or so it was thought. In 2013, a neuroscientist named Macon Nedergaard, working at the University of Rochester, published a paper describing what she called the glymphatic system. She had been injecting a tracer dye into the cerebrospinal fluid, the clear liquid that bathes the brain and spinal cord, of mice, and watching where it went. And she found that during sleep, the tracer spread rapidly throughout the brain, moving through spaces between cells and carrying waste products out to be cleared. During wakefulness, the spread was minimal. During sleep, particularly during slow-wave sleep, the tracer moved everywhere. The mechanism was extraordinary. Nedergaard found that during sleep, the cells of the brain physically shrink. Brain cells, astrocytes, the supporting cells of the neural tissue, contract by about 60%, expanding the spaces between cells from about 14% of brain volume to about 23%. With those spaces widened, cerebrospinal fluid can flow in, driven partly by the pulsations of blood vessels, washing through the brain tissue and carrying metabolic waste out through channels alongside draining veins. The brain, during sleep, takes out its own trash. Literally, the cells make room, the fluid floods in, the waste is carried out, and the cells expand again in the morning. Nedergaard's follow-up studies showed that amyloid beta was cleared from the mouse brain twice as fast during sleep as during wakefulness, and subsequent research has found evidence of the same system operating in human brains. The connection to Alzheimer's disease is not proven, but is deeply suggestive. Chronic sleep deprivation is associated with increased amyloid beta accumulation in the brain, faster progression to Alzheimer's pathology in genetically susceptible animals, and epidemiologically, with higher risk of cognitive decline. One of the most plausible mechanisms for why sleep deprivation is so bad for the brain over the long term is that it prevents adequate glymphatic clearance, and amyloid beta accumulates night by night, year by year, in brains that are consistently shortchanged on deep sleep. There is a particular irony in the Alzheimer's sleep connection that is worth sitting with. For decades, disrupted sleep was understood as a symptom of Alzheimer's disease, a consequence of the neurodegeneration rather than a contributor to it. Patients with Alzheimer's have well-documented sleep disturbances, particularly disruption of slow-wave sleep and circadian rhythm. The standard clinical assumption was the disease damages the brain regions that control sleep, and so sleep suffers, cause flowing one direction. What the glymphatic research suggests is that the causality may also flow the other way. Disrupted sleep reduces glymphatic clearance, which allows amyloid beta to accumulate, which damages the neurons that control sleep, which further disrupts sleep, which further impairs clearance. A vicious cycle, potentially beginning decades before any cognitive symptoms appear. The early stages of Alzheimer's pathology, amyloid accumulation in the brain, can be detected by brain imaging 20 to 30 years before clinical dementia develops. This is the window in which intervention matters most, and sleep may be one of the most accessible interventions available. Not a drug, not a surgical procedure, but an adequately long, adequately deep night of sleep, clearing the amyloid that would otherwise accumulate. This is speculative, and no clinical trial has yet established that improving sleep in middle-aged adults reduces their risk of Alzheimer's. But the biology is compelling enough that the question is being actively studied, and the answer matters for hundreds of millions of people. Just sit with that for a second. Your brain, while you sleep tonight, will physically shrink to make room for a flood of cerebrospinal fluid that will carry out the waste products of everything you thought about today. This is happening right now. The architecture of your brain is changing, temporarily, reversibly, purposefully, to enable a cleaning process that runs on the same schedule as your sleep cycle, that is deepest during the slow waves of stage 3, that cannot happen efficiently while you're awake. You are being cleaned from the inside while you're unconscious, and in the morning you'll wake up and drink coffee and never think about it. Now let's talk about REM sleep, because REM is where the most dramatic and the strangest things happen. REM sleep is paradoxical, and the word paradoxical is actually sometimes used as its scientific name, paradoxical sleep, because of its central contradiction. The brain in REM is, by almost every electrical measure, awake. The EEG looks like wakefulness. The neurons in the cortex are firing rapidly and irregularly, processing information, generating experience. Vivid, narrative, emotionally charged experience, the kind we call dreaming. The eyes are moving rapidly under closed lids, as if scanning some visual scene. The heart rate is irregular. Breathing is irregular. Brain temperature rises. Blood flow to the cortex increases. Every indication is that the brain is working intensely, perhaps harder than during wakefulness, and yet the body is completely paralyzed. Not just relaxed, paralyzed. During REM sleep, a specific circuit in the brainstem sends inhibitory signals to the spinal motor neurons, the cells that control voluntary muscle movement, and those neurons are actively suppressed. This is called REM atonia, and it is almost total. The postural muscles, the limb muscles, the muscles that would move your arms and legs if you were awake, they receive no signals. They cannot move. The only muscles exempt from this paralysis are the ones you don't need to suppress: the diaphragm, which continues to breathe, and the eye muscles, which produce the rapid eye movements that give REM its name. Why? The answer most sleep researchers accept is that REM atonia exists to keep you from acting out your dreams. During REM, the brain is generating movement commands. The experience of running, reaching, fighting, flying involves motor program activation just as real movement would, and without the atonia, those commands would translate into actual physical movement. You would be dangerous. You would be dangerous to yourself and to anyone near you. The evidence for this comes from cats. In the 1960s, a French neurophysiologist named Michel Jouvet discovered that if he made precise lesions in the brainstem region responsible for REM atonia in cats, the cats, during their REM periods, would stand up, stalk around, bat at invisible objects, appear to be chasing or fighting phantom prey, acting out, apparently, the content of their dreams while remaining technically asleep with the characteristic EEG of REM. Jouvet called the condition REM sleep without atonia, and it demonstrated both that the atonia was the result of an active inhibitory mechanism and that without it, the motor programs generated during dreaming would be executed. In humans, the equivalent condition is called REM sleep behavior disorder. People with REM sleep behavior disorder lack the atonia during REM and physically act out their dreams, shouting, punching, kicking, falling out of bed, sometimes injuring themselves or their bed partners. It is, by all accounts, frightening to witness and frightening to experience. And it is clinically important because it is, in a significant proportion of cases, a precursor to Parkinson's disease and other synucleinopathies. The same brainstem regions damaged in Parkinson's are the ones responsible for REM atonia, and REM sleep behavior disorder often precedes the other symptoms of Parkinson's by a decade or more. Sleep clinicians who see a patient with REM sleep behavior disorder now know that they may be watching the very early stages of a neurodegenerative disease. But in the intact, healthy brain, the atonia holds. And while the body lies still, the brain generates its most vivid experiences. Dreams have fascinated and baffled humanity for as long as humanity has been capable of being fascinated and baffled. Every culture that has left records has also left records of attempts to interpret dreams. Ancient Mesopotamians had dream interpreters in temples. The Egyptians had texts describing the meanings of specific dream images. The Greek Asclepieia, healing sanctuaries, practiced incubation, where the sick would sleep in the sanctuary, hoping for a healing dream from the gods. The Hebrew Bible, the New Testament, the Quran, all contain dreams of divine significance. Across cultures, across millennia, the dream was understood as a message from the gods, from the dead, from some part of the self that spoke only in the dark. The modern scientific study of dreams begins with Freud, or rather, begins with Freud's attempt to put dreams into a scientific framework that has largely not held up. Freud's 1900 work, The Interpretation of Dreams, argued that dreams were the royal road to the unconscious, that they were disguised expressions of wishes, primarily sexual and aggressive wishes, that were unacceptable to the waking conscious mind, and therefore represented in symbolic form during sleep. The manifest content of the dream, what you remember, was a distorted version of the latent content, the underlying wish. The purpose of dreaming, in Freud's system, was to allow these wishes a kind of release that prevented them from disturbing sleep. This is elegant, and it is largely unfalsifiable, and most current sleep researchers do not find it compelling as a scientific account of dreaming. But Freud did get some things right. He understood that dreams are not random noise. He understood that they have structure. He understood that the emotional content of dreams is not incidental. These intuitions have proven to be more durable than the specific theory. The most influential neuroscientific theory of dreaming came in 1977 from Harvard psychiatrists J. Allen Hobson and Robert McCarley. Their activation synthesis hypothesis proposed that dreams are the brain's attempt to make sense of essentially random activation during REM sleep. The brainstem, during REM, sends random bursts of activation to the cortex, particularly to the visual cortex and the motor cortex, and the cortex, being the meaning-making machine it is, tries to construct a coherent narrative from these random inputs. The dream is the story the cortex tells to explain the noise. In this view, dreams are not messages from the unconscious, but confabulations, post hoc narratives stitched together from random activation. The significance people find in their dreams is found, not sent. This theory, too, has critics, and it has been substantially revised. More recent models try to integrate both the bottom-up activation, Hobson and McCarley's brainstem signals, with top-down processes, the activation of emotional memories, the work of memory consolidation, the role of the default mode network. The picture that's emerging, still incomplete, is that dreaming is probably neither purely random nor purely purposeful in the Freudian sense, but something more complex. The brain using the opportunity of REM sleep to replay, recombine, and emotionally process the experience of waking life, generating narratives that are shaped by both the random firing of the brainstem and the existing structure of memory and emotion. The emotional intensity of dreams, the fear, the joy, the grief, the bizarre urgency of dream logic, is not incidental. The amygdala, the brain's emotional processing center, is one of the most consistently active regions during REM sleep. In waking life, the prefrontal cortex, the seat of rational deliberation, typically modulates the amygdala, putting emotional reactions into context, applying the brake of reason to the raw response of feeling. During REM sleep, the prefrontal cortex is substantially deactivated. The amygdala runs without its usual modulator. Emotions in dreams are not regulated in the way waking emotions are. They are raw, they are saturating. A dream of embarrassment feels more embarrassing than any waking embarrassment. A dream of falling feels like actual falling. The emotional experience is amplified rather than dampened. This disinhibition of the emotional system during REM has been proposed as essential to REM's function. The brain may need to reactivate emotional memories at full intensity in order to process them, to identify their significance, to extract the information they contain, to determine what to do with them. But doing this while the frontal cortex is offline, in a state where the stress hormone norepinephrine is absent, may create a unique condition: full emotional re-experience without full stress response. The memory is replayed at emotional intensity, but the fear circuit doesn't fully activate. Over repeated cycles of nocturnal replay under these conditions, the emotional charge of the memory is gradually separated from its informational content. What the memory means, the lesson, the consequence, the relationship to other experiences, is preserved and strengthened. The raw emotional pain of the experience is attenuated. This is, in essence, a theory of why we don't feel acute emotional distress about things that happened to us years ago. REM sleep has processed them, extracted the information, and filed it without the full emotional intensity of the original event. When this process is disrupted, as in PTSD, where the hyperarousal of the condition prevents the normal nocturnal emotional processing, the memories remain in their raw form. They are not processed. They are not attenuated. Every time they are retrieved with their original emotional intensity. The recurring nightmares of PTSD are not random. They are the brain trying, night after night, to accomplish the processing it cannot complete, because the arousal of the trauma keeps interrupting the REM that would do the processing. The nightmare is the failed attempt at repair. Prazosin, an alpha-1 blocker that reduces norepinephrine signaling, is one of the more effective pharmacological treatments for PTSD nightmares. The mechanism is exactly what the REM emotional processing theory would predict. By reducing norepinephrine during sleep, prazosin recreates something closer to the normal chemical environment of REM, the environment of low norepinephrine in which emotional memory processing is supposed to occur, and the nightmare frequency decreases. The connection between the drug's mechanism and the theory's prediction is not an accident. It is evidence. The emotional intensity of dreams, the fear, the joy, the grief, the bizarre urgency of dream logic is not incidental. The amygdala, the brain's emotional processing center, is one of the most consistently active regions during REM sleep. In waking life, the prefrontal cortex, the seat of rational deliberation, typically modulates the amygdala, putting emotional reactions into context, applying the brake of reason to raw feeling. During REM sleep, the prefrontal cortex is substantially deactivated. The amygdala runs without its usual moderator. Emotions in dreams are not regulated the way waking emotions are. They are raw, saturating. A dream of embarrassment feels more embarrassing than any waking embarrassment. A dream of falling carries real physical fear. The emotional experience is amplified rather than dampened. This disinhibition of the emotional system during REM has been proposed as essential to REM's function. The brain may need to reactivate emotional memories at full intensity in order to process them, to identify their significance, extract the information they contain, determine what to do with them. But doing this while the frontal cortex is offline, in a state where the stress hormone norepinephrine is absent, may create a unique condition, full emotional re-experience without full stress response. The memory is replayed at emotional intensity, but the fear circuit doesn't fully activate. Over repeated cycles of nocturnal replay under these conditions, the emotional charge of the memory is gradually separated from its informational content. What the memory means, the lesson, the consequence, the relationship to other experiences, is preserved and strengthened. The raw emotional pain is attenuated. Prazosin, an alpha-1 blocker that reduces norepinephrine signaling, is one of the more effective pharmacological treatments for PTSD nightmares. The mechanism is exactly what the REM emotional processing theory predicts. By reducing norepinephrine during sleep, it recreates something closer to the normal chemical environment of REM, the environment in which emotional memory processing is supposed to occur, and nightmare frequency decreases. This is not a coincidence. It is evidence that the theory is tracking something real. Matthew Walker, a neuroscientist at UC Berkeley and author of the book Why We Sleep, has described REM sleep as a kind of emotional first aid, a process during which the emotional content of recent memories is reactivated and gradually decontextualized, the raw experience being processed until what remains is the information content without the full emotional charge of the original event. This theory is supported by the observation that the amygdala, the brain's emotional processing center, is highly active during REM, and that the neurochemical environment of REM sleep is strikingly different from wakefulness. Norepinephrine, the brain's stress chemical, is essentially absent during REM. This chemical absence may create a safe environment for reprocessing emotionally charged memories, replaying them without the fear response that accompanied the original experience. People who are deprived of REM sleep show impaired emotional regulation. PTSD, many researchers now believe, involves a failure of REM sleep to adequately process traumatic memories. The memories keep reactivating in their full emotional intensity because the normal nocturnal processing was disrupted by the hyperarousal of PTSD itself. Walker's research also includes work on memory consolidation, the process by which new memories are stabilized and integrated into long-term storage. The basic finding, replicated many times in many labs, sleep after learning improves retention. Students who sleep after studying perform better on tests than students who stay awake between study and test, even when total time elapsed is matched. What's more, different stages of sleep appear to consolidate different types of memory. Slow-wave sleep, the deep stage we discussed earlier, seems most important for declarative memories. Facts, events, the kind of explicit knowledge you could state out loud. REM sleep seems most important for procedural memories, motor skills, pattern recognition, the kind of learning that happens when you practice something until it becomes automatic. A musician learning a difficult passage, a surgeon learning a new technique, an athlete mastering a movement. All of these depend in part on the REM sleep that follows practice, during which the motor program is somehow further consolidated in circuits the practitioner is not aware of. There is a specific phenomenon called targeted memory reactivation that has been studied in laboratory settings and that makes the memory consolidation function of sleep unusually concrete and tractable. In these experiments, participants learn material, spatial memory tasks, vocabulary, motor sequences, while being exposed to specific sounds or odors associated with what they're learning. Then, during sleep, the same sounds or odors are played softly, below the threshold of waking. The subjects don't consciously hear them, but during slow-wave sleep, the cues trigger replay of the associated memories in the hippocampus. You can see this in brain scans. And in the morning, memory for the cued material is better than for uncued material. You can, in essence, nudge the sleeping brain toward replaying specific memories by providing the same cues that were present during learning. The sleeping brain responds. It prioritizes. The consolidation is not random. This has obvious implications. Some researchers are exploring whether targeted memory reactivation could be used therapeutically to help people with anxiety disorders reprocess fear memories during sleep, or to help students consolidate studied material more efficiently. But the more fundamental point is what it reveals about what slow-wave sleep is doing. It is not simply dumping everything into long-term storage. It is selecting. It is replaying. It is active, organized, and responsive. And non-REM sleep, particularly slow-wave sleep, may do something even more interesting than simple consolidation. There is evidence that the brain, during slow-wave sleep, doesn't just replay memories, it selectively replays them, choosing which experiences to strengthen and which to weaken. There is evidence that during slow-wave sleep, the brain draws connections between newly learned material and the existing network of prior knowledge, finding analogies, making links, doing the kind of integrative thinking that we associate with insight and creativity. Some of the most famous examples of creative insight that occurred in sleep: the chemist Auguste Kekulé dreaming of a snake biting its own tail, and waking up understanding the ring structure of benzene, Paul McCartney dreaming the melody of Yesterday, Dmitri Mendeleev dreaming the arrangement of the periodic table. These may not be pure legend. They may reflect a real phenomenon, the brain, freed from the constraints of focused wakefulness, making connections it couldn't make while awake, and surfacing them as the dream images of the half-sleeping mind. Now, let's talk about the boundary places, because some of the most extraordinary things the sleeping body does happen not in the depth of sleep, but at the edges, the crossing from wakefulness into sleep and the crossing back. The state between waking and sleep, specifically the transition into sleep, stage 1, is called hypnagogia. And hypnagogia is, by any measure, one of the strangest states of consciousness available to an ordinary human being. As the brain relaxes from wakefulness toward sleep, the normal filtering mechanisms that keep perception orderly begin to loosen. Sensory impressions become vivid and unpredictable. Images appear, often geometric at first, then narrative and figurative, with a clarity and spontaneity that feels qualitatively different from ordinary imagination. Voices may be heard, single words, fragments of sentences, names called. Colors may be more vivid than anything available in waking vision. The experience is typically brief and is often not remembered. Artists and scientists have been aware of hypnagogia and have tried to harvest it for creative purposes. Salvador Dali is said to have developed a deliberate technique. He would sit in a chair holding a metal key over a plate and allow himself to fall asleep. The moment he crossed into sleep, his muscles would relax. The key would fall onto the plate with a clang, and he would wake in the hypnagogic state with whatever image he'd been holding in his mind still present and available. Thomas Edison reportedly used a similar technique with steel balls instead of a key. Both were trying to capture the hypnagogic state's particular quality of image, uninhibited, combinatory, strange, and bring it back into waking creative work. Whether or not the specific anecdotes are accurate in their details, the underlying strategy is consistent with what we know about hypnagogic imagery. It is the product of a brain state that is genuinely different from ordinary imagination and genuinely interesting to people who care about unusual mental content. The equivalent at the other boundary, coming out of sleep rather than going in, is called hypnopompia. Hypnopompic experiences tend to be less rich and varied than hypnagogic ones, and they often shade into a different and more distressing phenomenon, sleep paralysis. Sleep paralysis occurs when you become conscious, or partially conscious, while the REM atonia is still in effect. You wake up, or you think you've woken up, but you cannot move. Your muscles will not respond. You can breathe, and usually you can open your eyes, but you cannot move your arms or legs or turn your head. The experience lasts from a few seconds to a few minutes, and ends when the atonia finally releases, either because your brain has crossed fully back into wakefulness or because the REM episode ends naturally. In the moment, it feels much longer. Sleep paralysis can occur in isolation, just the paralysis, disorienting but not terrifying. Or it can occur with hallucinations. The hallucinations of sleep paralysis tend to be distinctive. They often involve a presence, a feeling, sometimes very specific, that someone or something is in the room with you. Often the feeling resolves into a visual hallucination, a shadowy figure in the doorway, a dark shape near the bed, a face at the window, and often, most distressingly, a sensation of pressure on the chest, as if something were sitting on you, pressing down, making it difficult to breathe. This cluster of experiences, paralysis, presence, pressure, visual hallucination, is so consistent across cultures and across history that it has generated its own folklore in essentially every human society. The English word nightmare originally referred not to a bad dream, but to this specific entity, the mare, a malevolent spirit that sat on sleeping people and caused distress. In Newfoundland folklore, it is the old hag. In German, the Alp. In Japanese, the kanashibari. In Mexican folklore, a dead person sitting on the chest. In West African traditions, a witch or spirit. In virtually every culture that has described sleep paralysis, and there is no culture that has not, the same elements appear: the paralysis, the presence, the pressure, the terror. A universal human experience generated by a specific neurological event interpreted through local mythology. Sleep paralysis is common. Estimates suggest roughly 8% of people experience it at some point in their lives, and it is particularly associated with irregular sleep schedules, sleep deprivation, and stress. It is not dangerous, though it is frightening, and knowing what it is, knowing that it is REM atonia, bleeding into waking consciousness, that it will pass, that the presence and pressure are hallucinations generated by a brain half in REM, can transform the experience from terrifying to merely strange. Let's talk about temperature, because temperature regulation during sleep is one of its most underappreciated and practically important aspects. Before we get to temperature, let's take a moment with the clock itself, because everything we're about to talk about, the temperature cycle, the melatonin rise, the cortisol surge at dawn, is coordinated by a clock, a literal biological clock, ticking in your brain, keeping time through a molecular mechanism that was so surprising and so elegant when it was finally worked out that it earned 3 American scientists the Nobel Prize in Physiology or Medicine in 2017. The clock is located in a tiny region of the hypothalamus called the suprachiasmatic nucleus, or SCN, named for its position just above the optic chiasm, where the optic nerves from the two eyes cross. The SCN is small, about 20,000 neurons in each hemisphere, 40,000 total. It receives direct input from specialized light-sensitive cells in the retina, cells that respond specifically to blue wavelength light and that project directly to the SCN, not to the visual cortex. Their entire job is to report the ambient light level to the clock, so the clock can stay synchronized with the 24-hour cycle of the external world. The SCN keeps time through a molecular feedback loop. The story of how this loop was worked out is one of the great detective stories of modern biology. It begins in 1971, when Seymour Benzer and Ronald Konopka, working with fruit flies at Caltech, identified a single gene whose mutation caused the fly's circadian rhythms to be disrupted. Some mutants had shortened cycles, some had lengthened ones, some had no detectable rhythm at all. The gene was called period. It encoded a protein called PER, but how PER kept time was unknown for another 15 years. The answer came from Jeff Hall and Michael Rosbash at Brandeis University and Michael Young at Rockefeller University, working through the 1980s and 1990s. They discovered that the period gene produces PER protein, and PER protein accumulates in the cell until it reaches a concentration high enough to enter the nucleus and turn off its own gene. As PER production stops, PER degrades. Its concentration falls below the threshold needed to suppress its own gene. The gene turns on again. PER is produced again, accumulates again, turns itself off again. This negative feedback loop cycles, with a period of approximately 24 hours. The clock is molecular. It runs by proteins suppressing their own synthesis, degrading, and allowing synthesis to restart. An elegant, self-contained oscillator, present in virtually every cell in the body, ticking reliably at a period close to 1 day. Hall, Rosbash, and Young shared the Nobel Prize in 2017 for this discovery. In their Nobel lectures, all three emphasized that the molecular clock is not a curiosity, it is universal, it is fundamental, and its disruption has consequences for virtually every aspect of health. Shift workers, who chronically work against their biological clocks, have significantly elevated rates of metabolic syndrome, diabetes, cardiovascular disease, and certain cancers. The circadian disruption caused by shift work is now listed by the World Health Organization as a probable carcinogen. The body's clock is not just about sleep, it is about the timing of everything: metabolism, immune function, cell division, DNA repair. Disrupt the clock, and all of these processes become desynchronized from the external world and from each other, with consequences that accumulate over years. Your core body temperature follows a circadian rhythm, a 24-hour cycle coordinated by your brain's internal clock. During the day, temperature rises. In the late afternoon and early evening, it reaches its daily peak, typically around 37.5 degrees Celsius. Then, as evening progresses toward your normal bedtime, it begins to fall. It drops through the night, reaching its low point in the early morning hours, a few hours before you typically wake. Then it rises again, helping to prepare your body for waking. The drop in core temperature at night is not a passive consequence of sleeping. It is an active process. Your body, in the evening hours, begins to radiate heat from its surface, particularly from the hands and feet, which have specialized blood vessels that open wide to allow rapid heat loss. Your skin temperature rises as your core temperature falls. The blood vessels near the surface of your hands and feet dilate, flushing warm blood to the surface, where it can radiate heat into the room. This is why a warm bath before bed can help you sleep, not because it warms you up, but because when you get out of the bath, Heat radiates rapidly from your dilated surface vessels, accelerating the drop in core temperature that promotes sleep onset. Wearing socks to bed has actually been shown to help some people fall asleep faster, for the same reason. Keeping the feet warm facilitates the peripheral vasodilation that dumps heat and lowers core temperature. The link between temperature and sleep onset is mediated by the same brain region that controls much of sleep, the preoptic area of the hypothalamus. This small region simultaneously drives body temperature down and drives sleepiness up. The two things, temperature falling and sleepiness rising, are not just correlated. They are mechanistically connected. Neurons in the preoptic area that fire to promote sleep are the same neurons that drive peripheral vasodilation. Cooling the preoptic area directly in animals induces sleep almost immediately. Warming it wakes them up. The sleep switch and the thermostat are the same switch. But before we get to temperature's stranger REM-related behavior, let's stay with REM for a moment and ask a question that people have wondered about for decades. When your eyes move rapidly during REM, are they tracking the scenes of your dream? The evidence is suggestive, but not conclusive. In one clever set of experiments, researchers induced lucid dreaming, the state in which a sleeper is aware they are dreaming and can control the dream to some extent, in trained subjects, and arranged a pre-sleep signal. Before sleep, the subjects agreed that during a lucid dream, they would move their eyes in a specific, pre-arranged pattern to signal that they were conscious and dreaming, and they did. EEG confirmed REM sleep, eyes moving in the agreed pattern, followed by reports of lucid dreaming when woken. The eye movements corresponded to what the dreamers reported looking at in the dream. This suggests that the eye movements of REM are, at least in part, meaningful, that the visual cortex is generating something like a visual scene, and the eyes are scanning it, just as they would scan a real visual scene in waking. The theater of the sleeping mind has a stage, and the eyes are watching it. This remains an area of active research and genuine uncertainty. The eye movements of REM are generated by brainstem circuits, and there is debate about how precisely they track dream imagery. But the convergence of evidence, the EEG patterns that resemble waking visual processing, the activation of the visual cortex during vivid dreaming, the studies with lucid dreamers showing correspondence between eye movements and reported gaze direction, makes the pure, random noise interpretation of REM eye movements increasingly difficult to maintain. And then, when REM sleep begins, something stranger happens. During REM, the brain largely stops regulating body temperature. The thermoregulatory systems that keep your core temperature in range throughout NREM sleep, the shivering, the sweating, the cardiovascular adjustments, are suspended during REM. You become, temporarily, what biologists call poikilothermic, an organism whose body temperature tracks the environment rather than actively defending a set point. Like a lizard in the sun, you warm when your environment warms and cool when it cools. If the room is cold, you'll get colder during REM. If the room is warm, you'll warm up. This is one reason why sleeping environments that are too hot are disruptive. During REM, you can't regulate yourself, and a hot room means a hot brain, which shortens or disrupts REM episodes. Cool rooms, around 18 degrees Celsius, are consistently associated with better sleep quality, particularly more and better REM sleep. Now, let's talk about hormones, because the night is not chemically quiet even when the body is still. The most famous sleep hormone is melatonin. Melatonin is produced by the pineal gland, a small, pine cone-shaped structure deep in the brain that Descartes once believed was the seat of the soul, and it is the body's primary signal for darkness, for night, for the appropriate time to sleep. Its discovery is a fairly recent story. The hormone was isolated and chemically identified in 1958 by an American physician named Aaron Lerner, working at Yale. Lerner had spent years trying to identify the substance that caused color changes in frog skin, a substance that earlier researchers had found in extracts of pineal gland tissue. When he finally isolated it, he named it melatonin, from the Greek melanos for dark. Melatonin production begins to rise in the evening, about 2 hours before your habitual sleep time. It peaks in the middle of the night and then falls toward morning, declining before you wake. It does not cause sleep directly. Melatonin is not a sedative in the pharmacological sense, but it signals to the body that darkness has arrived and that sleep is appropriate. It lowers core body temperature, as part of the cascade we just described. It suppresses alertness. It prepares the body's physiology for the shift from wakefulness to sleep. And melatonin is exquisitely sensitive to light, specifically to blue wavelength light, the kind emitted by the sun during the day, and also, unfortunately, the kind emitted by the screens of phones, tablets, and computers. Even brief exposure to blue light in the evening can suppress melatonin production for hours, shifting the body's sense of when night has arrived and delaying sleep onset. This is not a trivial effect. Studies consistently show that screen exposure in the hour or two before bed can shift sleep timing by 30 minutes to an hour, can reduce melatonin levels substantially, can reduce REM sleep duration, and can leave people feeling less rested the next morning. The technology that we use most heavily in the hours before bed is, at a physiological level, telling our pineal glands that it's still afternoon. While melatonin is rising in the evening, another hormone is falling, cortisol. Cortisol is the body's primary stress hormone, released by the adrenal glands in response to physical or psychological stress, but also following a circadian rhythm that is independent of stress. During the day, cortisol maintains alertness, mobilizes energy, keeps immune responses calibrated. At night, cortisol falls to its lowest point. Then, in the early morning hours, around 3 or 4 a.m., often before you're aware of being awake, cortisol begins to surge. By the time your alarm goes off, cortisol levels are approaching their daily peak, having been rising for 2 to 3 hours. This cortisol surge is part of what wakes you up. It mobilizes blood sugar, raises blood pressure, increases alertness, essentially primes your cardiovascular and metabolic systems for the demands of the day ahead. This cortisol awakening response is one of the reasons that being woken abruptly by an alarm at a time your body hasn't yet prepared for feels so genuinely awful. Your cortisol hasn't peaked yet. Your core temperature is still near its nadir. The physiological preparation for waking hasn't completed. You are being pulled out of sleep before the sleep has finished what it was doing. The grogginess of a bad alarm waking, sleep inertia it's called, is a real physiological state, the consequence of abrupt transition from deep sleep or late REM to wakefulness before the cortisol awakening response has fully run. The hormonal landscape of the night extends well beyond melatonin and cortisol. Testosterone, in men, is largely secreted during sleep, particularly during REM sleep. The first morning testosterone level, measured on waking, is substantially higher than levels measured in the afternoon. This is not a minor variation. Nocturnal testosterone secretion accounts for most of the daily production. Men who are chronically sleep-deprived show testosterone levels equivalent to men 10 years older. A week of sleeping 5 hours per night instead of 8 reduces daytime testosterone levels by 15 to 20%. This has effects on energy, mood, libido, and muscle maintenance. It is one of the reasons that sleep is considered one of the most potent performance-enhancing strategies available to athletes. Two other hormones that deserve mention are leptin and ghrelin. The the hormones that regulate hunger and satiety. Leptin, produced by fat cells, signals fullness to the brain. When leptin is high, you feel satiated, you eat less. Ghrelin, produced by the stomach, signals hunger. When ghrelin is high, you feel hungry, you eat more. Sleep deprivation systematically shifts both. Leptin falls, ghrelin rises. The sleep-deprived person is hormonally primed to feel hungrier and less satisfied after eating. Studies consistently find that people who sleep less eat more calories, more high-carbohydrate foods, more late-night eating. The effect size is not trivial. Subjects in controlled sleep restriction studies consume, on average, about 250 to 300 extra calories per day compared to when sleeping adequately. Over a year, that difference would produce meaningful weight gain. The relationship between sleep deprivation and obesity is almost certainly mediated in part by these hormonal shifts. The sleep-deprived person is not eating more because of weak willpower. They are eating more because their hormones are telling them they are hungry and that food is not enough. The cardiovascular changes during sleep are subtle but worth understanding because they have real clinical importance. In the early stages of NREM sleep, heart rate slows and blood pressure falls. By slow-wave sleep, both are substantially lower than waking values. This nocturnal dip in blood pressure, typically 10 to 20% below daytime levels, is an important part of cardiovascular health. People who fail to show this nocturnal dip, called non-dippers, have significantly higher rates of heart disease, stroke, kidney damage, and overall cardiovascular mortality than people who dip normally. The nighttime pressure dip appears to give the cardiovascular system a critical period of reduced workload and reduced mechanical stress. Without it, the vessels and the heart age faster. Then comes REM, and the dip reverses. Heart rate and blood pressure become irregular during REM, reflecting the autonomic storms of a dreaming brain, and can show brief, sharp increases. These REM-associated surges are normally benign, but in people with underlying heart disease, they can be significant. The highest risk period for heart attacks is the early morning hours, roughly 3 to 9 a.m., which corresponds to the period of maximum late-night REM sleep combined with the rising cortisol of the pre-waking phase. It is not a coincidence. The cardiovascular system during late-night REM is the cardiovascular system under autonomic stress. For a compromised heart, that stress can be the trigger. One of the most underappreciated aspects of the sleep cardiovascular connection is what happens to heart rate variability, the beat-to-beat variation in the time between heartbeats. In wakefulness and light sleep, heart rate variability is high. The heart speeds up and slows down responsively with each breath, with changes in posture, with fluctuations in blood pressure. This variability is a sign of a healthy, responsive autonomic nervous system. During deep, slow-wave sleep, heart rate variability decreases. The heart beats in a steadier, more regular pattern, reflecting the reduced autonomic demands and the cardiovascular system's deeply rested state. During REM, the pattern changes again. Variability increases sharply and erratically, reflecting the autonomic storms of the dreaming brain. The heart in REM is doing something that looks, on a beat-by-beat basis, almost like mild exercise, not the resting, stable rhythm of quiet wakefulness, but an irregular, variable pattern driven by the emotional and motor activation of dreaming. For people with structurally normal hearts, this is not a problem. For people with arrhythmias or reduced cardiac reserve, the autonomic variability of REM sleep can be the trigger for events that would not occur during calmer stages. This is why both sudden cardiac death and cardiac arrhythmias show a strong circadian pattern, with peaks in the early morning hours when late-night REM is at its longest. The heart that was safe through the night becomes vulnerable during the last REM episode before waking. Breathing during sleep changes in ways that are more consequential for more people than almost any other sleep-related phenomenon. In wakefulness, breathing is controlled by both voluntary and automatic mechanisms. You can choose to breathe fast or slow, to hold your breath or take a deep one, but you'll also breathe perfectly well without thinking about it at all. In sleep, voluntary control is suspended. Breathing is entirely automatic, driven by brainstem circuits that respond to blood oxygen and carbon dioxide levels. This automatic control works well in most people most of the time. But the transition to sleep brings a specific vulnerability. The muscles of the upper airway, the tongue, the soft palate, the walls of the throat, relax in sleep just as every other muscle relaxes. In people whose upper airways are narrower than average, whether from anatomy, from excess soft tissue, from nasal congestion, from sleeping position, this relaxation can cause the airway to narrow enough that airflow becomes turbulent. That turbulence is the sound of snoring. Snoring, in mild form, is simply noise, unpleasant for bed partners, not medically significant in itself. But in people with obstructive sleep apnea, the airway doesn't just narrow. It collapses. The walls of the throat seal together, and airflow stops entirely. The sleeper, who is unaware of this, continues trying to breathe against a closed airway, chest and belly heaving, until the oxygen level in the blood falls low enough to trigger an arousal. The brain wakes partially, the airway tone increases, the airway reopens, often with a gasp or snort, and breathing resumes. Then the cycle begins again. This can happen 5 times an hour in mild cases, or 60 or more times an hour in severe cases. The person never reaches deep sleep, or REM. Their blood oxygen dips repeatedly. Their blood pressure surges with each arousal. Their heart works harder than it should all night, every night. The treatment that changed the lives of millions of people with obstructive sleep apnea was invented in 1981 by an Australian physician named Colin Sullivan, working in Sydney. Sullivan had been studying sleep apnea and understood the physics of the problem. The airway was collapsing because the pressure inside it, during the inward effort of a breath, became negative relative to the surrounding tissue, pulling the walls together. If you could maintain a slightly elevated air pressure inside the airway, enough to push the walls apart and prevent collapse, you could prevent the obstruction. The device that applies this gentle, continuous positive pressure to the airway through a mask worn over the nose during sleep is called a CPAP machine, continuous positive airway pressure. Sullivan's first prototype, built in his laboratory, used a vacuum cleaner motor run in reverse, pushing air rather than pulling it. The mask was a rubber diving mask sealed around the edges with silicone. His first patient was a man with severe sleep apnea who hadn't had a restorative night of sleep in years. Sullivan connected the device, the man slept, and within minutes was in deep, slow-wave sleep, and then REM, stages of sleep he hadn't been reaching for so long that the transition was immediately visible on the EEG recordings Sullivan was monitoring. The man woke up and said he hadn't felt that rested in years. CPAP today is sleeker and quieter than a vacuum cleaner motor in a mask, but the principle is identical, and the benefits extend well beyond sleep quality. CPAP treatment in people with significant sleep apnea reduces blood pressure, reduces the risk of heart attack and stroke, improves glucose metabolism, reduces daytime sleepiness, and in observational studies is associated with substantially reduced mortality. What sleep apnea was doing, night after night, was slowly eroding cardiovascular function. What CPAP does is give the cardiovascular system back its nightly rest. Now let's talk about the thing that happens at the very beginning of falling asleep, that almost everyone has experienced, and that almost nobody can explain when asked about it. You're drifting off. Your thoughts are loosening. You're in that comfortable, soft-edged state between waking and sleep. And then, suddenly, you feel a jerking sensation. Your leg kicks out, or your whole body lurches, and you're wide awake, heart beating slightly faster, perhaps feeling slightly foolish if you're sharing a bed. The sensation is often accompanied by a brief visual image, tripping, falling, missing a step, and then it's gone, and you drift off again. This is called a hypnic jerk, or a sleep start, or hypnagogic myoclonus, and it happens to roughly 70% of people. Its physiological cause is not fully settled. One explanation is that it is a byproduct of the transition between wakefulness and sleep, when the brainstem is recalibrating its control of muscle tone, a brief misfire in the motor system as it shifts from waking mode to sleeping mode. Another explanation, more speculative, but popular because it has a satisfying evolutionary logic, is that the hypnic jerk is a vestigial reflex inherited from primate ancestors who slept in trees. The logic goes, as a tree-sleeping primate drifts toward sleep and loses muscle tone, the brain might interpret the slackening as the beginning of a fall, and reflexively jerk the muscles to catch itself. If this is right, the startling sensation of falling that accompanies the hypnic jerk is literally the brain of an arboreal ancestor living in your brainstem, catching you in a tree that doesn't exist. There is something I want to come back to about sleep spindles, because they're one of the features of stage 2 sleep that has attracted the most research attention, and for good reason. Sleep spindles, those brief bursts of 11 to 16 Hz oscillations that appear in the stage 2 EEG, are not just noise suppressors. They appear to play an active role in memory consolidation, particularly for factual and declarative memory. The spindles seem to coordinate the transfer of information from the hippocampus, where new memories are initially stored, to the cortex, where they're eventually integrated into long-term storage. The hippocampus is a temporary holding area. It can store new information quickly, but its capacity is limited, and it was never designed for permanent storage. The neocortex has vast capacity but learns slowly. Memory consolidation, in the current understanding, involves the hippocampus replaying new memories during sleep, and the cortex gradually learning from those replays, the spindles pacing and synchronizing the transfer. People who generate more sleep spindles tend to score better on memory tests the following day. There is variation in spindle density that appears to be partly genetic, and that correlates with general intelligence in some studies, a finding that has attracted attention and some controversy. What is more clearly established is that specific manipulations can increase spindle density and improve next-day memory performance. Auditory stimulation at certain frequencies during stage 2 sleep increases spindle density, and the memory improvement correlates with the spindle increase. The spindle is not just a byproduct of stage 2. It is doing something, and what it is doing appears to matter. Let's talk about the immune system, because sleep and immunity are connected in ways that are far more direct and far more consequential than most people realize. During sleep, particularly during slow-wave sleep, certain immune processes are upregulated. The production and release of cytokines, the chemical messengers of the immune system, increases. Natural killer cells, the immune cells responsible for destroying virally infected cells and cancer cells, are more active after adequate sleep than after sleep deprivation. Naive T cells, the cells that can be educated to recognize new threats, migrate more efficiently to lymph nodes during sleep, because the hormonal environment of sleep, low adrenaline, reduced cortisol, promotes the adhesion molecules that allow these cells to enter lymphoid tissue. The immune system uses sleep, in a sense, to do its administrative work, to mount and coordinate responses, to consolidate immunological memory of past infections, to prepare for future challenges. The clinical evidence for this is striking. A study published in 2015 by researchers at USF deliberately exposed participants to rhinovirus, the common cold, and monitored who developed symptoms. People who had been sleeping less than 6 hours per night in the weeks before exposure were 4 times more likely to develop a cold than people who had been sleeping 7 hours or more. Not somewhat more likely, 4 times. Sleep deprivation is one of the most powerful known predictors of susceptibility to the common cold. The effect size rivals that of the most significant known risk factors. Perhaps the most dramatic demonstration of sleep's role in immunity comes from a study of influenza vaccination. A research team gave influenza vaccines to 2 groups, one group that slept normally after vaccination, and one that was kept awake for the night after receiving the vaccine. When the researchers measured antibody levels 10 days later, the sleep-deprived group had produced less than half the antibodies of the normally sleeping group. The vaccine, in the absence of sleep, was significantly less effective. The immune system needs sleep to mount a full response to an antigen, to consolidate the immunological memory of the vaccine. This finding has direct implications for real-world vaccination programs, and it suggests that getting a good night's sleep before and after vaccination is not just good hygiene, but good immunology. Matthew Walker, in summarizing a decade of research on sleep and the immune system, described sleep deprivation as one of the most powerful factors in suppressing immune function. He noted that even one night of sleeping just 4 to 5 hours reduces natural killer cell activity by roughly 70% compared to a full night's sleep. The immune system, in the context of truncated sleep, is running at a fraction of its normal capacity. This connection goes in both directions. Not only does sleep deprivation impair immune function, immune activation causes sleep. When you're sick, you sleep more. Cytokines released during infection, particularly interleukin-1 and tumor necrosis factor, act directly on the brain to promote sleep. The fatigue and sleepiness of illness are not incidental annoyances, they are deliberate. The immune system is telling the brain to sleep more, because sleep is when the immune system works best, and you are sick and it needs all the help it can get. Sleep is the immune system's operating environment of choice. Let's spend a moment with the animals, because the variety of sleep behavior across the animal kingdom is one of the things that most strongly argues for sleep being not just important, but essential. Not a luxury, but a biological necessity, so fundamental that evolution has found a way to preserve it, even in circumstances where its cost seems prohibitive. Dolphins and porpoises, and many other cetaceans, practice what is called unihemispheric sleep. They sleep one hemisphere of the brain at a time. One half of the brain goes into sleep mode, generating slow waves, while the other half remains awake, controlling breathing, maintaining muscle tone, monitoring the environment. The eye connected to the sleeping hemisphere closes. The eye connected to the awake hemisphere remains open. The dolphin continues to swim, continues to surface for breath, continues to detect threats, all while half of its brain is sleeping. After some time, the hemispheres switch. The previously sleeping hemisphere wakes, the previously waking one sleeps. And so the animal gets its sleep without ever being fully unconscious, without ever being fully vulnerable. This is not sleep in the sense that you and I sleep, and yet it is clearly sleep, producing the same slow-wave electrical signatures, serving what appears to be some of the same restorative functions. The need is apparently so strong that dolphins evolved an extraordinary solution to the problem of sleeping while needing to breathe, swim, and stay alert. Rather than giving up sleep, they invented a way to sleep that doesn't require giving up the other things. Birds face a similar challenge during long migrations. The Arctic tern migrates from the Arctic to the Antarctic and back, a round trip of roughly 70,000 kilometers, twice a year. During this migration, it needs to sleep. It cannot sleep on the water safely. It cannot stop. A 2016 study by a team led by Niels Rattenborg used accelerometers implanted in frigate birds to directly monitor their brain activity during long oceanic flights, and found that the birds showed clear EEG signatures of both slow-wave sleep and, briefly, REM sleep while airborne. They slept for short bursts during flight, particularly the hemisphere contralateral to the inside of a banked tern, while the other hemisphere kept the bird on course. They slept, cumulatively, about 41 minutes per day during long overwater flights, a tiny fraction of the 10 hours they typically slept when on land, but enough, apparently, to sustain them. Sleep, even reduced to its minimum, was maintained. Sleep changes dramatically across the human lifespan, and the changes are not arbitrary. They reflect the different biological priorities of different life stages. Newborns sleep 16 to 17 hours a day. They have no consolidated nocturnal sleep period. They sleep in multiple short episodes distributed throughout the 24 hours because their circadian clock has not yet been entrained to the light-dark cycle. The SCN is present and ticking at birth, but it takes several months for the connections from the retina to mature enough to effectively synchronize the clock to external light. This is why new parents describe feeling like they've lost track of night and day. Their baby genuinely doesn't know the difference yet. More strikingly, roughly 50% of newborn sleep time is in REM sleep, or more precisely, in a state called active sleep, which has some of the characteristics of adult REM. Adults spend about 20% of their sleep in REM. Infants spend 50%, and fetuses, in the womb, may spend 80% of their sleep time in REM-like active sleep. This developmental pattern, peak REM in early life, declining through childhood and adolescence to adult levels, suggests that REM sleep plays a particularly important role in brain development. The amount of time the developing brain spends in active sleep tracks closely with the period of most intense synaptogenesis, the formation of new neural connections. REM sleep, in early life, may provide the endogenous stimulation that drives the formation and pruning of synaptic circuits in the developing cortex. The dreaming brain of an infant may literally be constructing itself. Adolescents experience a well-documented circadian phase shift during puberty. The circadian clock shifts forward toward later timing during adolescence, such that the biological drive for sleep pushes later into the night and the biological morning is later too. An adolescent whose biological clock says it's 7 a.m. when the school day demands they wake at 6:30 is not lazy. They are biologically early. The hormonal changes of puberty directly affect the circadian clock, and the evidence is robust enough that dozens of school districts in the United States have experimented with later start times for high schools, with positive effects on attendance, grades, mental health, and even car accident rates among teen drivers. In older adults, the architecture of sleep changes in the opposite direction. Slow-wave sleep decreases, sometimes dramatically. A 60-year-old may get only a fraction of the deep sleep they had at 20. The timing of sleep advances. Older adults tend toward earlier bedtimes and earlier wake times, the reverse of adolescence. Sleep becomes more fragmented, with more brief arousals throughout the night. The ability to achieve and maintain deep, slow-wave sleep declines because the neurons in the brain regions that generate those slow oscillations decline with age. Whether this decline in slow-wave sleep contributes to the cognitive changes of aging specifically to the impaired memory consolidation that is one of the earliest and most consistent findings of normal cognitive aging, is an area of active research. The hypothesis is compelling. If slow-wave sleep is essential for memory consolidation, and slow-wave sleep declines with age, then the decline of memory with age may be partly a story about declining sleep quality. And if you could restore deep sleep in older adults through carefully designed acoustic or electrical stimulation that entrains the slow oscillations, you might also restore some of the memory consolidation capacity. This is being tested. The results so far are cautiously promising. Sleep changes dramatically across the human lifespan, and the changes reflect the different biological priorities of different life stages. Newborns sleep 16 to 17 hours a day. They have no consolidated nocturnal sleep period. They sleep in multiple short episodes distributed around the clock, because their circadian clock has not yet been entrained to the light-dark cycle. The SCN is present and ticking at birth, but it takes several months for the retinal connections that synchronize it to mature. This is why new parents describe losing track of night and day. Their baby genuinely doesn't know the difference yet. More strikingly, roughly 50% of newborn sleep time is in REM sleep, or a state called active sleep with similar characteristics. Adults spend about 20% of their sleep in REM. Infants spend 50%, and fetuses in the womb may spend 80% of their sleep time in REM-like active sleep. This developmental pattern, peak REM in early life, declining through childhood and adolescence to adult levels, strongly suggests that REM plays a central role in brain development. The amount of time the developing brain spends in active sleep tracks closely with the period of most intense synaptogenesis, the formation of new neural connections. The dreaming brain of an infant may literally be constructing itself. Adolescents experience a well-documented circadian phase shift during puberty. The clock shifts forward, toward later timing, such that the biological drive for sleep pushes later into the night. An adolescent whose biology says wake at 8 a.m. isn't lazy when school demands 6:30. They are biologically early. The hormonal changes of puberty directly affect the circadian clock, and school districts that have shifted to later start times for high schools consistently find improved attendance, grades, mental health, and lower teen driver accident rates. In older adults, slow-wave sleep decreases, sometimes dramatically. A 60-year-old may get only a fraction of the deep sleep they had at 20. The neurons that generate slow oscillations in the prefrontal cortex decline with age. Whether this decline in slow-wave sleep contributes to the memory changes of aging is an area of active research. The hypothesis is compelling. If slow-wave sleep is essential for declarative memory consolidation and slow-wave sleep declines with age, then some of the memory difficulty of normal aging may be a story about sleep, not just neurons. And if you could restore slow-wave sleep in older adults through acoustic or electrical stimulation that entrains the slow oscillations, you might restore some of the memory consolidation capacity. This is being tested. The results so far are cautiously encouraging. The great white shark does not sleep in any way that has been observed. It must keep moving to oxygenate its gills, and traditional sleep postures are incompatible with perpetual motion. Insects appear to have sleep-like states. Even fruit flies have rest states that resemble sleep, that are regulated by some of the same genes and neurotransmitters, and that show the same consequence of deprivation that mammalian sleep shows. Rest-deprived fruit flies perform worse on learning tasks and die sooner. The evolutionary distance between a fruit fly and a human is roughly 600 million years, and yet both need something that looks like sleep. Whatever sleep is doing, and we'll get to the full complexity of that question, it is so important that it has been conserved across nearly 1 billion years of animal evolution. Now let's talk about what happens when sleep is lost, because the consequences of sleep deprivation are among the most well documented in all of biology, and they are sobering in ways that should matter to everyone who routinely shortchanges their sleep. In July 1964, a 17-year-old high school student in San Diego named Randy Gardner, motivated partly by scientific curiosity and partly by the desire to set a record, stayed awake for 11 days and 25 minutes, 264 hours, under medical supervision. He is still the record holder for the longest documented stint without sleep by a young, healthy person under controlled conditions. His experience has been extensively documented. The first night was easy. By day 2, Gardner was noticing difficulty with coordination and concentration. By day 3, he was experiencing mood changes, irritability, then brief periods of profound depression. By day 4, he was hallucinating. Not vague impressions, but specific hallucinations. He looked at a street sign and saw a person standing there. He thought he was a famous football player. By day 6, his speech was slurred, and he had difficulty completing sentences. By day 10, his memory was fragmented, his face expressionless. He could not concentrate on anything for more than a few seconds. By day 11, he had to be led around to prevent him from walking into things. He was awake, but what was left of ordinary consciousness was barely recognizable as such. Gardner slept after the record and recovered. Sleep deprivation of this acute, dramatic kind does not leave permanent damage in young, healthy people. The brain, given the opportunity, rebounds. In the first recovery sleep, Gardner slept for 14 hours and 50 minutes. He then slept a normal amount the second night and subsequent nights. He did not pay back his 11 days of sleep debt in one enormous marathon. The brain is not a simple bank account. You cannot fully repay sleep debt by simply depositing double the usual amount. What recovery sleep does is prioritize. The first recovery night is disproportionately rich in slow-wave sleep, as the brain rushes to accomplish the deep sleep-dependent restorative processes it was denied. REM rebounds on the following nights. The recovery is orderly, but not complete. The military has spent enormous resources studying performance under sleep deprivation because military operations routinely demand sustained wakefulness. The findings are consistent and sobering. After 17 hours awake, cognitive performance is equivalent to that of someone with a blood alcohol level of 0.05, slightly below the legal limit in many jurisdictions. After 24 hours awake, performance equivalency reaches a blood alcohol level of 0.10, well above the legal limit. The same operations a surgeon performs competently after a good night's sleep become measurably less precise, less safe, after a long overnight shift. The same driving maneuver a person executes reliably after 8 hours of sleep becomes genuinely dangerous after 20 hours awake. And the tragedy, or perhaps the insidious feature, is that people in this state reliably underestimate their impairment. They feel okay. They believe they are performing adequately. The very faculty needed to judge impairment, clear metacognition, accurate self-assessment, is itself one of the first things that sleep deprivation degrades. There is a broader point here that is worth stating plainly. Sleep is not a weakness. It is not wasted time. It is not something that productive, driven, high-performing people can and should minimize in the service of getting more done. The evidence is overwhelming and unambiguous. Sleep is when performance is built. The athlete who trains hard but sleeps poorly will recover more slowly, build less muscle, develop sharper reflexes more slowly than the athlete who trains equally hard and sleeps well. The surgeon who operates on 5 hours of sleep makes more errors than the surgeon who operates on 8. The student who pulls an all-nighter before an exam remembers less the next morning than the student who studied for fewer hours but slept. Sleep is not the enemy of achievement. It is one of its prerequisites. Gardner slept after the record and recovered. Sleep deprivation of this kind, dramatic and terrible as it is in the moment, does not leave permanent damage in young, healthy people. But the military and aviation industries have spent decades studying the consequences of more chronic, more moderate sleep restriction, the kind that millions of people experience voluntarily, sleeping 6 hours when they need 8, and the results are consistent and alarming. Cognitive performance, reaction time, decision-making, emotional regulation, working memory, all decline predictably with each night of suboptimal sleep, and they decline in a way that is not subjectively apparent. People who have been sleeping 6 hours a night for 2 weeks perform, on cognitive tests, as badly as people who have been awake for 24 hours straight. But they don't feel that impaired. They report feeling fine, or nearly fine. They lose track of how impaired they've become. This is one of the most dangerous features of sleep deprivation, the progressive undermining of the very faculties needed to recognize the impairment. There is a condition that takes this to its terminal extreme. Fatal familial insomnia is a prion disease, caused by a misfolded protein, like mad cow disease, that targets the thalamus, the brain's sleep relay, and causes progressive insomnia. The condition begins in midlife, with worsening sleep disturbance and strange anxiety. Within months, the patient is barely sleeping at all. The thalamus deteriorates. Hallucinations become continuous. Motor control is lost. Dementia develops. Death follows, typically within 12 to 18 months of the first symptoms. It is invariably fatal. There is no treatment, and it has been described by patients' families as one of the most terrible deaths imaginable. A person watches themselves lose their mind while being unable to sleep, while knowing exactly what is happening. Fatal familial insomnia is vanishingly rare. Fewer than 60 families in the world carry the mutation, but it demonstrates, in the starkest possible terms, what it looks like when sleep becomes permanently, irreversibly unavailable. Let's talk about one more thing before we arrive at tonight, and that is the question of dreams we remember versus dreams we forget. Most dreams are forgotten within minutes of waking, sometimes within seconds. You wake, you sense the fading tail of a dream, and then it is gone before you can bring it into focus. Some people remember many dreams, others claim never to dream at all. Though research consistently shows they are dreaming, they simply aren't waking during REM periods when recall would be possible. The memory of a dream depends on waking from it, or very close to it, because the hippocampus, which is the brain's first memory-forming structure, has reduced activity during REM, and without hippocampal encoding in the minutes following the dream, the experience simply doesn't transfer into any storable form. This forgetting is probably not a flaw. If you retained full, detailed memories of every dream you'd ever had, the boundary between experience and imagination would be extremely difficult to maintain. The brain, it seems, is designed to use the content of REM and forget the wrapper, to extract whatever processing benefit the dream served, without cluttering waking memory with thousands of vivid but fictitious experiences. The forgetting is part of the function. The exception is when you wake during a REM period or very shortly after one, and have a few minutes of quiet lying still before fully entering wakefulness. In those minutes, the dream can sometimes be captured, recalled in enough detail to hold on to. Whether this is worth doing is a personal question. Dream journals exist. Some people find the practice valuable for creative work or for emotional awareness. Others find it simply delays the transition to wakefulness without obvious benefit. What is consistent is that active effort, in the first minutes after waking, is required to retain what the dreaming brain produced. Let's talk about one more thing before the why, the question of which dreams we remember and why we forget most of them. Most dreams evaporate within minutes of waking, sometimes within seconds. You surface, you sense the tale of something, and then it's gone before you can name it. Some people remember dreams often. Others claim never to dream at all. Though research consistently shows they are dreaming, they simply aren't waking during or after REM, when recall would be possible. The memory of a dream depends on waking from it, or very close to it, because the hippocampus, the brain's first memory-forming structure, has reduced activity during REM, and without hippocampal encoding, in the moments following the dream, the experience simply cannot transfer into any storable form. Dreams live in the present tense of the sleeping mind. The moment that mind transitions to something else, they are gone. This forgetting is probably not a flaw. If you retained full, detailed memories of every dream you'd ever had, the boundary between experience and imagination would be extremely difficult to maintain. You would carry thousands of vivid but entirely fictitious events as apparent memories. The brain, it seems, is designed to use the processing that dreams provide and discard the wrapper, to extract whatever the dream served without cluttering waking memory with its content. The forgetting is part of the function. There is a practice called dream journaling, keeping a notebook by the bed and writing down whatever you can capture in the first moments of waking before the hippocampus has finished releasing the dream. Some people find this valuable for creative work or emotional awareness. What consistently happens, if you do it regularly, is that your dream recall improves, not because you're dreaming more, but because you're developing the habit of catching the content in the window when it's still catchable. What you find, typically, is that your dreams are far stranger and richer and more emotionally charged than your waking memory would have suggested. The unconscious, at night, is working hard. It just prefers not to be remembered. So why does sleep exist? We've talked about what it does: the memory consolidation, the glymphatic clearing, the immune function, the hormone pulses, the emotional processing. But the honest scientific answer to why sleep exists is that we still don't fully understand it. It is remarkable to say, given how universal sleep is and how much research has been done, but sleep remains in many ways mysterious. There is no single consensus theory that accounts for everything sleep does. The energy conservation theory, sleep as a way to reduce metabolic demand during the unproductive hours of darkness, when finding food is difficult, has some support, but can't account for the large amount of metabolic activity during REM, or for why predators sleep as much as prey species. The memory consolidation and synaptic homeostasis theories have strong empirical support, particularly for slow-wave sleep, but don't fully explain REM. The immune function theory is well supported, but seems unlikely to be the whole story. The emotional processing theory is compelling, particularly for REM, but lacks the mechanistic grounding that the glymphatic hypothesis has for slow-wave sleep. The most honest position is probably this: sleep is doing multiple things simultaneously, different stages doing different things, and we have partial accounts of most of them. The glymphatic hypothesis for slow-wave sleep may be one of the most mechanistically grounded. The observation that brain cells physically shrink to allow CSF influx during sleep is a structural, measurable fact, and the consequences for amyloid clearance are quantifiable. But even here, the full story is not told. What we do know with certainty is that sleep is essential. The synaptic homeostasis hypothesis, developed by Giulio Tononi and Chiara Cirelli at the University of Wisconsin, is one of the most influential current theories of sleep function. The hypothesis proposes that waking experience, learning, perceiving, thinking, strengthens synaptic connections in the brain. The more you experience, the stronger your synapses become. But synaptic strength cannot keep increasing indefinitely without cost. Stronger synapses require more energy to maintain, take up more space, consume more metabolic resources. If every waking experience left a permanent strengthening trace, the brain would eventually run out of dynamic range. There would be no room to learn new things because all the synaptic capacity would already be committed to old things. Sleep, in Tononi and Chiarelli's view, is when the brain scales down synaptic strength globally, weakening all connections slightly, restoring the metabolic and computational resources needed for the next day's learning. This is called synaptic downscaling, and there is direct experimental evidence for it. Measures of synaptic strength in mice are higher at the end of the waking day and lower after sleep. The slow oscillations of deep sleep appear to be the mechanism of downscaling, each oscillation involving a period of near silence across the cortex, during which net downward pressure on synaptic weights is applied. This theory predicts that the memories that survive sleep are the ones that started strongest, the ones encoded most deeply during the day, or reinforced by repetition or emotion. The weakest memories, the ones formed incidentally and never consolidated, are lost during the overnight downscaling. The ones that mattered most to the brain, by whatever measure of mattering the hippocampus applies, survive. Sleep is not just a time to copy memories into permanent storage. It is also a time to edit, to decide, by the logic of synaptic strength, what was worth keeping and what was not. Animals deprived of sleep die, in some animal models, from sleep deprivation alone, faster than from food deprivation. Every tissue in the body depends on sleep. Every cognitive function is impaired by its absence. Every organism complex enough to have a nervous system appears to have some version of it. Whatever it is doing something that nothing else can substitute for. There is no drug, no meditation, no brief rest, no amount of coffee that can replicate what happens in the sleeping brain and body. The nightly loss of consciousness is not a waste of a third of your life. It is the maintenance schedule that makes the other 2/3 possible. Let's end where we began, because I want you to think about what's happening right now, in this moment, as you lie here and these words become less and less distinct in your awareness. Your melatonin has been rising for the past hour or two. Your core body temperature has been falling. The vessels in your hands and feet have opened wide, radiating the heat that the night is taking from you. Your cortisol is at its daily low. Somewhere in the preoptic area of your hypothalamus, neurons that simultaneously promote sleep and drive temperature down are firing with increasing frequency. The threshold between waking and sleeping is very close. When you cross it, in the next few minutes, or perhaps you already have, and these words are reaching you through the soft filter of stage 1, your brain will begin its work. The slow, rolling eye movements of stage 1 will start. The sleep spindles of stage 2 will follow, gating out the small sounds of the house, the cars outside, the settling of walls, so that none of it reaches you. The threshold is guarded. You are being protected from waking. Then the slow waves will come. The neurons of your prefrontal cortex will begin to oscillate together, great sheets of them cycling between firing and silence, and those cycles will propagate across the cortex in slow rolling waves. Your brain cells will begin to shrink, fractionally, measurably, purposefully, and the cerebrospinal fluid will begin to move into the spaces they've opened. The waste from everything you thought about today, the amyloid beta and the tau protein, and the other metabolic byproducts of all that neural activity, will begin its transit. Your pituitary gland will release its nightly pulse of growth hormone, and in muscles and bones and skin and organs, the slow work of cellular repair will begin. The day's damage addressed. The proteins that need replacing flagged for renewal. Your immune system will shift into its maintenance mode. Cytokines will rise. Natural killer cells will activate. The immunological memory of past infections will be tended. If you were vaccinated recently, the antibody response will be consolidated in a way it cannot be during wakefulness. The body is using the quiet hours for the work that wakefulness interrupts. And then, sometime in the middle of the night, the first REM period will arrive. The brainstem will issue its command to the motor neurons, the gentle, total, protective paralysis. The amygdala will come alive. The prefrontal cortex will go dark. Norepinephrine will fall to nearly zero. And in that particular chemistry of low stress and high activation, whatever needs processing from today will be processed. Memories will replay. Connections will be drawn. The emotional weight of things will be quietly adjusted, not erased, but integrated, made livable, made part of the story rather than an interruption of it. You will not know any of this is happening. It will happen without your awareness, without your participation, without your consent or effort. This is the most extraordinary thing about it. The most sophisticated biological process you have access to, the nightly reconstruction of your mind and body, requires nothing from you except the willingness to let go. You don't have to do it right. You don't have to try to sleep correctly. You just have to stop trying to stay awake. The rest is handled, and these words are reaching you through the soft filter of stage 1. Your brain will begin its work. Stage 2 will claim you first, and sleep spindles will begin to fire, protecting you from the small sounds of the night. Then the slow waves will come. Your brain cells will begin to shrink. The cerebrospinal fluid will begin to move. The waste from everything you thought about today will begin its slow transit toward drainage. Your pituitary will release its pulse of growth hormone, and the cells of your body will begin the repairs that the day's demands accumulated. Somewhere in the middle of the night, in the first of what will be 4 or 5 cycles, you will enter REM for the first time. Your brainstem will send the signal that paralyzes your voluntary muscles, all except the diaphragm and the small muscles of the eye. Your brain will become as electrically active as wakefulness. The amygdala will come alive. Norepinephrine will fall to nearly zero. And in that neurochemical state, highly active, highly emotional, free of the stress chemical that normally moderates the intensity of experience, you will begin to process the emotional weight of your day, replaying and recontextualizing the things that mattered, filing them into the long structure of memory in forms that will be more usable tomorrow than they were this evening. Your immune system is preparing. Your cells are rebuilding. Your memories are consolidating. The day that just ended is being woven into the fabric of who you are. You don't have to do anything. You don't have to try. This is the extraordinary thing about sleep. All of this happens without effort, without intention, without awareness. The brain that does all of this does not need your permission or your participation. It has been doing it every night since the night you were born, in the same sequence, through the same cycles, toward the same restoration. It is the most reliable thing your body does. So let the words go soft. Let the images come loose. Let the boundary between waking and sleeping become as permeable as it wants to be. The thoughts that feel important right now will still be there in the morning, probably clearer than they are at this moment, because your sleeping brain will have spent the night rearranging them into something more useful. Whatever happened today, whatever you're carrying into the dark, your brain is already beginning to do what it always does, sorting through it, making sense of it, cleaning up after it. You just have to let go. Rest well.